I have the pleasure of introducing Dr. Guerrero, specializing in advanced imaging technologies, focusing on their applications at the nano and micro scale. He serves as a researcher at the National Laboratory for Advanced Microscopy at the National Autonomous University of Mexico, contributing to the emerging field of optical nanoscopy. His work centers on the extensive development and application of super resolution microscopy techniques, as well as providing deeper insight into the molecular architecture of the natural world. Committed to international collaboration, Adan emphasizes the importance of educational equality and universal access to higher education, as it's a catalyst for both scientific and societal advancement. In his role, he concentrates on developing comprehensive educational programs in bioimaging, particularly aiming to nurture scientific communities in Mexico and Latin America. I'm really excited to hear you talk today. Single frames, super resolution microscopy, is it a myth? I don't know. I'm ready to find out. So please take it away. Thank you very much.
Okay, can I start sharing my screen right now? Fantastic. So I'm going right now. Oh, yeah. So can you see my screen? Great.
Looks great.
Okay, let me just make smaller all the windows. Okay. Well, today I'm going to speak about this concept, which is single frame super resolution microscopy. So it comes from a field that is not really emergent at all, because it started in the 60s when people was trying to study the space and to decompose the information that satellites were collecting. But before, let me say something about where we are coming from. This is related to the identity. So we come from Mexico, from the National Laboratory for Advanced Microscopy. So this is one of the first optical microscopy centers open to everybody, regardless if they are academics, if they come from the commercial or even artistic sectors. So we opened in 2013, and then after we create other labs in the north part of Mexico, here in Ensenada. Can you see my cursor? Yes. And also later on, we start another LNMA in a medical center in Mexico City, one of the biggest medical centers in Mexico. And so now we are a network of microscopy experts, and we try to really make cohesion in Mexico through different programs. For example, the Mexican Bioimaging Workshops is happening right now. And we are trying to bring education to several colleagues in Mexico. So in the lab, or in these networks of labs, we are experts in microscopy, mostly optical, but also electron microscopy. So we provide service, training, coaching, dissemination, education, and also outreach. I'm part of the group that do indeed research and development, and we really are concentrated on creating bonds between all the communities, so within Mexico and abroad. So in our lab specifically, we have expertise in plenty optical microscope techniques, from confocal, total internal reflection microscopy, FCS, SPEM, super resolution microscopy, and so on and so forth. Today, I will speak about some technological advances that we have done in the field of super resolution microscopy. So, and the main focus of today is to understand nature at the nanoscales. And we all know that optical microscopes are limited by diffraction, and this limits the quantity of information that we can get when this information comes from the nano scales. And when we are trying to understand subcellular regimes like subcellular organelles or even viral particles or proteins. So we are always limited by diffraction, and it is very difficult to extract information out there. And this is because we live in a blue representation of reality. And this is not only in optical microscopy, but it is in all the reality. And I'm depicting this with these images in which we can have an object, a real object, that has certain degree of detail. But as the information travels through an information channel, it just becomes convoluted, and it will end up having some blur. And this blurring relates the capacity that we have to describe what is in there. So when we speak in optical microscopy, this happens, but also happens in any instance of imaging. For example, here, I'm just representing what happens to an image, and when this information becomes convoluted, when this information becomes transformed. And I have here two ways of representing the real image and the Fourier transformation of this image. Because so if we do the Fourier transformation of this image, here in the center, we will have the lower frequencies, and in the periphery, we will have the higher frequencies. And to exemplify what means by lower and higher frequencies, here, what happens if we set all this information to 0? So if we set all the information related to the higher frequency, we actually see a blur. And this is what it seems that it's happening in optical microscopy, that there is information that we cannot see, and that's why the images become blurred. But what happens if we put a mask here and we put all the information in 0 in the center? Well, when we invert the transform, we will get the edges. Okay, so there is a way to control how much information is being transmitted, and if information is going through a channel, let's say that this channel is a microscope, it can be filtered out. Okay, and this is what we are seeing here, that we have a real object, and the photons or the information that is emanated from this object becomes convoluted within the optics of the system, and we can see the information in the back focal plane of the lens, which is the Fourier transformation, or we can even project this information in a camera that will create the image. So, when we speak about information and information transfer, so we will need to understand what is the optical transfer function in optical microscopy, which is defined as the Fourier transform of the point express function that describes how different frequency components travel and are modulated through the optical system. And when we think in Fourier, a lens or a microscope or imaging collection system has a capability to transmit information, and this is called as the optical transfer function, where the numerical aperture of the lens creates like a mask, like a finite aperture that ends up what we know is that ends up defining the maximal resolution of a system. So if we think about resolution, this was an idea that starts more than 200 years ago by Epps, and he defined the resolution limit in optical microscopy, and this resolution limit sets a boundary to the maximal observable frequency. So in this case, if we see the Fourier transformation of an image, in the center we'll have the lower frequencies, which is the low contrast, which is the bigger objects, and far away we have the high frequencies, which might resemble to the texture. We can see here that there is a limit, and this limit is defined by the numerical aperture of the lens. And after this limit, we will have either zeros or noise. And this is the known resolution limit in optical microscopy expressed in the Fourier spectrum. So why I'm speaking all about this complexity of Fourier and so on and so forth? Because this process, as it has a mathematical counterpart and a physical counterpart, it might be possible to reverse it. And this is the role that the convolution has done in humanity, trying to reverse the convolution process. So let's think. So here we have an object, and in the bottom part, I'm putting the Fourier transformation and the notation in the Fourier space. So here we have the real image, and here we have the notation that will go to the Fourier transformation. So this object becomes convoluted with the point express function of the instrument, that in Fourier space is the multiplication of the object by the Fourier transformation of the point express function. This will give us the image. Okay, this is the image that we collect with our eyes when we are looking at the APs of the microscopes, or is the image that we are collecting in the camera, which is also convoluted with noise. So what the deconvolution process tries to do is to recover the object. And look here, if we see in the Fourier space, the operation becomes very easy in the sense that we just move this OTF term to this point, and then we will try to divide our Fourier transformation of our image between the Fourier transformation of the optical system. Then we might want to recover the object. This is the convolution. But as I already told you here, if we have the optical transfer function that is collected through a microscope, here we have zeros, 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 meaning that if we divide anything by zero, what we have no definition. And this is an important aspect because we cannot recover more information beyond the diffraction limit with the convolution, with this operation, because everything out here is zero or undefined. That is the limit of the convolution. And that is what I was teach about the convolution, that you can correct some of the deformation that an imaging system produces into your data, but you cannot surpass the diffraction limit. Well, that is not true at all, because people that was working trying to understand the information that was collected from the space used numerical approximation like using Bayesian statistics to reconstruct this problem. And there is a very well-known algorithm, which is the Richardson-Lucy algorithm for the convolution, which is one of the most used algorithms for the convolution. Mm-hmm. And in here, I'm just showing an original object. And in the bottom, we have the Fourier transformation of this object. So if we just put a mask here to remove the higher frequencies, we will end up having some blurring. And if we try to solve the simple equation like this of the convolution, we will not recover the information. But in the case of Lucy Richardson and other approximations, what you end up having is actually recovering all of that information. What? But the information wasn't lost. Yes, it was not lost. It was not lost, just that the information gets transformed. And this is very interesting because what actually had set up the capability of the convolution algorithms is not the mathematical principles, it's noise. And let me show you an example. So this is a modeling in which these guys were just put two lines, and they separate the lines each certain distances. And they convolve with a point spread function of about 90 nanometers. Then, as you can see, we lose the capability to solve the parallel lines. But if we do Lucy Richardson, look at this. We can recover the information, almost everything. This is a model, and it will be great if we can do this with any image that we collect with a single microscope. But the problem is this: data is corrupted by noise. So then the problem becomes ill-defined. So in this case, the original object had noise, Poisson and Gaussian, and it is convoluted with the same PSF. What you can see here that after Lucy Richardson, we are not recovering all the information. And what happens in this procedure is that actually Lucy Richardson is an iterative procedure that tries to model the system and tries to create an approximation of what is the reality. But when you have noise, the noise corrupts the data, and then it creates artifacts. That's why people is not using in practice the convolution like Lucy Richardson to extend the resolution of images beyond the diffraction limit. So there is new technology that is just appearing in this area, in which people have to really plug into the questions more information about the system. So in this case, our colleagues model the problem like if you have an image is a collection of sparse information, like information that comes from fluorophores, which is also continuous, and the continuity is given by the diffraction. So by solving these functionals in which people try to approximate to the real solution, and they want to recover the object. So they compute the difference between the image and the object, which is corrupted by noise. They assume some priors of continuity and sparsity. And what it actually happened is that in this work, Sao and colleagues approximate to the problem to the point that they can double the resolution in optical microscopy, which is great because, as I already told you, the solution in the maths was very, very early in the 60s. And then after Lucy, Richardson, Hahn, and other colleagues were able to approximate it, but noise was limiting the solution. But nowadays we have introduced more information to the questions, more priors about information, and then we managed to duplicate the resolution. And this is a recent paper. But let's really give credit to the people that have started this way of thinking. And in this paper that was published in the 90s by Carrington and colleagues, so they also approached the problem by modeling, in which they have an image which is corrupted by noise, and they model the system considering the camera, the information, and in this case the information was continuous, was finite amount of data, like photons that are digitalized in a camera, but this information comes from a continuous object, a continuous object, which might be a cell. And what they try to solve is to compute the difference between the data and the model using some empirical stabilization terms for the model to actually works. And what they have is that in this case, they model one single point or 2 points, and then after they convolve with the PSF, they got an image which was digitalized and modeled with the noise of the camera. And then after using this model, they managed to get the solution. So this was the initial model. Here we have a single fluorophore. Here we have 2, and that is the solution that they come out. So what we are seeing here is that if you understand enough of the system, you can model it, and then you can do single frame super resolution imaging. Okay, and here I showed you some of the data in which these people took images of cells and with microtubules, and they managed to compensate some of these problems of diffraction. So, but why the field of super resolution microscopy has really taken over through the instrumental solutions that try to drive the fluorophores to the blinking or to the darkest states? Well, it seems that in all this sense, we have to model, and we need to know enough information of the system to actually understand it. So then after, we will not have a general solution. So what I'm going to present today is how we end up with that general solution. But first of all, let me just summarize that the light is a electromagnetic wave that has properties of waves, but also have properties of particles. I told you that if we think in the Fourier optics, we are considering all the properties of light as electromagnetic waves, and that's why we end up with this idea of convolution of information. But also, light flows as particles. And in this case, the particles might be the photons that are being produced from fluorophores, and those photons are collected by the lenses. And which is very, very interesting is that photons emanate from, in the case of optical microscopy or fluorescence microscopy, let's simplify the problem, photons emanate from single sources, which will be the fluorophores. Okay, so if we think about the problem from the diffraction from the diffraction aspect. Here we have that the light travels through the optics, and we end up having this uncertainty, and the uncertainty is related to diffraction that set up the diffraction boundary. But if we see the information as particle, then the shape of the distribution has no role, because then we can think in a problem in which what we seek is to find the source of information. So, and then we will now start to approach the problem from the particle nature of light. Okay, so let me set up the problem. In this cartoon, we have a distribution of information where we really don't care about the shape of the distribution, but we want to understand where is the source of information. So what we can do is we will use an approach that comes from statistics, that we will start in a certain point of this space, and we will do local comparisons, and then we will compute a vector that will point to the maximal source of information. So this is called the mean shift approach. So what the mean shift does is that it starts in a given point, calculate the mean without considering the central value, and that then computes the difference. What we end up having is a vector, and as you can see, this vector points to the source of information, and you can do an iterative process to find the source. The mean shift has been previously used to identify edges, like to track particles, to track objects. But there is another property that was not really explored before, which is the magnitude. As you can see, the magnitude of this vector becomes smaller as soon you approach. Then it might be possible to reduce the uncertainty just by computing the magnitude of this vector. And this is what we actually did. So what we have here is that, let's think that we have a Gaussian distribution, but this is a general solution. And in this case, if we compute the mean shift, we can reduce the uncertainty. So this is called the mean shift super-resolution principle, and it was developed by Slade Torres, a PhD student in my lab. And the idea is just like that. So here, you can compute the mean shift, and you can get the magnitude of the vector. And if we compute the inverse of that vector, we will have something like a Mexican hat. And this will help us to reduce the blurring effect caused by diffraction. So then I will explain here some theoretical considerations. So let's imagine that we have a Gaussian distribution, which is the simplest approximation to the diffraction problem. So we compute the mean shift. We have something like a Mexican hat. We invert the Mexican hat, and there we have it. And then after, we remove the negative values, and we multiply this distribution by the original. As you can see, the distribution becomes shrinked. So this is called measure of zero order. And what we can do now is start from measure zero, and we can subtract measure zero from the initial image. And then we get this distribution. If we invert this distribution, we get a second Mexican hat. Then we multiply this distribution by this one, and we remove all the values of the tails and compensate by the initial intensity. As you can see here, measure order zero use a single Mexican hat to increase resolution. Measure of order one use another Mexican hat to increase resolution even more. If we describe this process in two dimensions, what we can have is each time that we compute a new Mexican hat, we increase the resolution more and more and more. And this is something that it is done instrumentally with a STED microscopy that creates a donut that removes information in the borders. So we create this digital donut, which is an inverse Mexican hat that increase even more and more the resolution. Of course, there is always certain information that we are losing. As we perform higher iterations of measure, we lose information of details. So this has to be bear in mind. And I normally use measure of order 0 and measure of order 1 because I really want to keep the much information as possible from my data. Okay, so to demonstrate this, let's first do the first, which is let's define what is the limit that we want to surpass. I already talked to you about the Abe's limit, but there are other limits that are related to information. And here I will speak about the sparrow limit, which is the sparrow, which is the limit where you don't see this dip, this valley in the distribution that is shaped by two united distribution. So if these two distributions are Gaussians, the sparrow limit is at two times their standard deviation. Okay, and here you will not be able to recognize that there is a single entity or two entities. Okay, so now we will repeat the same procedure of measure, but considering this distribution created by two joint distributions at the sparrow limit. What you can see here, after computing measure of order 0, we actually can separate these two individual emitters. By using measure of higher orders, we can we refine much more the information and we manage to separate it. But wait. If we think on information and compute the size of this valley, if the valley goes to 0, if the amplitude goes to 0, that means that we can really separate the fluorophores. And if the amplitude is 1, that means that you cannot separate it. This is called the dip. Okay, so in this case, the dip, 1 means not resolved, and 0 means fully resolved. And here you have the Rayleigh limit and the sparrow limit. As you can see here, when we start to approaching the fluorophores from far distances to the same position, so this is the limit that was defined by Rayleigh. This is the sparrow limit, which is the information limit. And it seems that we have reached a new theoretical limit for measure, which is actually 1.6 sigma, which is more or less double resolution. Here, at this limit, you can see that we can actually separate the fluorophore depending in the iteration order of measure. Okay, but this is just a theoretical approximation, and we don't know if we had found a novel limit of resolution. Then we went to perform some experimental demonstrations. So in this case, here we have the GATA SIM nanoruler, which is a small chain of DNA that have fluorophores in the nitrogen bases. And this nanoruler is a commercial proof that you can use to test the resolution in your optical system. So we buy them, and then we perform structural illumination microscopy, and we actually saw that we can solve the nanorulers. But what it is very interesting is this. So this is the diffraction limit of a couple of nanorulers. As you can see, there is no way to decide to conclude that there is a single or there are any rulers there. But after we perform the measure at single frame, we managed to really actually solve the nanorulers. So this is an experimental demonstration that measure can solve objects that are below the diffraction limit. So then after we went to test the Argo light pattern, which is created by laser lithography. And here you can see two lines of fluorophores, which are separated at different distances, like the model that I already showed with the convolution. So here, the lines are separated either 0, 30, 60, 120, 240, or 390 nanometers. Here you can see the Rayleigh limit for confocal microscopy. And what you can see here is that at the Rayleigh limit, we can really separate the fluorophores. But what happened here, where you cannot conclude that there are two lines of fluorophores? Well, if you process the same information by measure, then you can actually see that we can duplicate the resolution of an optical system. I mean, in this case, the confocal, the resolution of a confocal microscope. Okay, but what happens if we use another system, which is the area scan system that use an array of detectors to collect much more information from the PSF. So in the area scan system, the resolution that we measured was 180 nanometers, and we managed to really improve this resolution. But at 100 of 20 nanometers, the area scan system was not able to solve these two lines of fluorophores. But by using MESR plus area scan, we can increase the resolution there, and we can conclude that there are two lines of fluorophores. So there is much more that can be achieved by MESR, but at the moment I will just focus in just duplicating the resolution. Okay, so then we test MESR in combination with structural illumination microscopy. Here we have two meiotic chromosomes, and here we have the sister chromatids. And as you can see in the diffraction limit data, you cannot either say that there are two chromatids, but after processing with SIM, you can see an increase of resolution, and with MESR, a higher increase. Okay, so what I'm trying to say is that MESR has the possibility of extend resolution in confocal microscopy, in area scan, in structural illumination microscopy. But what happened if we combine MESR with other technologies like STED microscopy? So in this image, what we have is immunofluorescence to detect acetylation or methylation in chromatin by detecting this post-translational modification in histones. Okay, so green means active chromatin, and inferno, which is between red and orange, means inactive chromatin. So if we see this confocal image, there is no way to determine that the chromatin might be either active or inactive or a combination. But if we do a STED, it is easy to see that there are territories of information. There are some areas in this nucleus that have inactive chromatin, and there are some areas that have inactive chromatin. So let's say that we are considering in Mexico, that you are in Mexico or in certain parts of Latin America where you don't have access to an STED instrument. So then you might have access to a confocal microscope or to an epifluorescence microscope. And what happens if you collect a confocal image and then after you do MESR? Well, what you can see from this data is that you can reach the same conclusion about the biology in this specimen, which is that we can see territories of, inactive or territories of active chromatin. So meaning that we can provide access to super resolution microscopy with technology that is at the hands of people in Latin America. Okay, but what happens if we combine technologies? So in this case, I'm just giving a zoom to what you can do with the STED by visualizing the same area that was visualized in the confocal mode. And what happens if we combine STED with MESR? Well, it is very interesting because in this case, in the STED image, you can actually see that there are areas of inactive and areas of inactive chromatin. But when you combine STED with MESR, there is no single spot of colocalization, meaning that in this experiment, chromatin can be in two states, inactive or active. That's it. So then MESR not only increase the resolution in regular microscopy, but it can increase the resolution and works together with other super resolution techniques. So now I will show you another way of doing super resolution, which is analyzing the temporal dynamics of information. So in this case, I already showed you that we can increase resolution of a single image using MESR, like kind of duplicating that resolution. But if we do a pixel-wise temporal transformation, which is, let's just compute the variance of a single pixel, and from that information, so the variance have information about the blinking of the fluorophores, then we can increase even more the resolution. And here I'm going to show a demonstration. what we have built here, those are called nano rulers. So what we have is a change of synthetic DNA, which was built in the lab, that was targeted that was developed to bring the CRISPR-Cas system. So in this case, we built a single RNA guide, which was extended to have an hybridization with a fluorophore, with a fluorescence proof. So what we have here is the Cas12 targeted to the DNA, which was separated each 100 nanometers. And then after we put a way to have association or dissociation of fluorescence proof. So in these experiments, what we are doing is atomic force microscopy just to measure the height. And what we can see here is the DNA. And here these two bonds are proteins bound to the DNA. And this distance is about 100 nanometers because we engineer these rules to have 100 nanometers. But what we have here is the nano ruler, and you will see blinking. The blinking will be created by association and dissociation of these fluorophores. So this is turf microscopy. And the experiment is paint, CRISPR paint. Okay, so when we collect single diffraction limited images, they look like this. We have the blinking, and we can refine any of these images, image by image with MESSER, and then we get images like this. Look that there is a reduction of noise, but also the emission of single detected spot also becomes reduced in the sense of a spatial distribution, and then we perform a temporal analysis. What happens is that we manage to increase the resolution a lot. So here we have the computation of the distances measured from this kind of experiments. And as you can see, the distance corresponds more or less to the engineered information. They are not perfect because the DNA is not like a straight ruler, but it falls and unfolds. So then after we went to test what would be the maximal resolution that we can achieve only with diffraction limited image and doing temporal analysis. So we managed to get these paint nano rulers that have sites of binding for fluorophores like those of paint, and that have the possibility to blink. So in this case, the nano rulers were engineered to have binding sites each 40 nanometers. And as you can see, after collecting 300 images from the same scene and analyzing each of them with single frame MESSER, and then after doing the variance transformation, we can solve them. Okay, so with this experimental say, we can conclude that by using MESSER and using a temporal analysis, we can reach a resolution of about 40 nanometers. So let's go to the analysis of single images. And in this case, I'm showing a video of fluorescence microscopy, and then you can see how much contrast we can gain with MESSER. But it's not only a gain of contrast, but it's a gain of resolution. And let's just stop a bit here and think about this. So I start this talk speaking about deconvolution, and that if you do the convolution can give you, can increase contrast, but not beyond the resolution limit. So there is a possibility to gain much more information from the data, but you have to understand more or less the data and perform some modeling, and then you can overcome the diffraction boundary. So through numerical approximations. What I just bring to the table here is that MESSER have the capability of increase the resolution of any image collected with using a fluorescence microscopy, and the solution is always the same. It is deterministic, and you don't need to know much information from your data, okay, which is very, very important. So regarding scientific discoveries, so here we use method to reproduce some findings that we actually did before with other super resolution approaches. So in this case, what we are seeing here are viral replication machineries. And in this paper, we demonstrate that these viral replication machineries are organized like onions, like concentric layers of viral proteins. And as you can see here, after processing the data, so in this case, we collect 100 images and we process them with temporal measure, you can see that it is possible to underscore the nano organization of these viral replication machineries. And you can do it by analyzing a temporal series of data, but also using a single frame. Okay, look at this case or this case. So as MESR can deliver nanoscopic detail from single images, so the maximal resolution in terms of time is defined by the experimental system that records the data. So then you can do live cell in super resolution microscopy and understand cell physiology at the nanoscales using MESR. Also, you can really analyze the data collected with instruments that allow deep tissue imaging. In this case, I'm only showing an image collected with regular confocal microscopy. So this is a root of Arabidopsis thaliana, and what we are seeing here is the mitotic division. So in this case, this image was collected prior to the existence of MESR, and I asked Hector Torres to test what will be the effect of doing MESR of the 3D volumetric information. Which is interesting here is that the pixel size was 500 nanometers. Indeed, it was not oversampled. But as you can see here, MESR actually managed to increase the contrast. But what happened if we repeat this experiment and collect the images with much more sampling? In this case, the data was collected with a pixel size of 50 nanometers, and the step size was also 50 nanometers in C. So what you can see here is that this is now the chromatin, and as you can see here, we have the chromosomes. This is the regular confocal max intensity projection, and here we can see what MESR does to this data. These little dots are nucleosome clutches. So meaning that if you have access to a confocal instrument, you can do 3D volumetric imaging, and then you can use MESR to study the nanoscopic organization without the need of having a high-resolution instrument. If you have a high-resolution instrument like an STED, then you can go and reach the molecular scales. So this is a reconstruction, and it is important here to tell you that this implementation of MESR is 2D. So we haven't released the implementation in 3D, but we are working on that. And here I just give you some examples, some technologies that we have test MESR. It is compatible with light sheet microscopy, confocal laser scanning microscopy, epifluorescence microscopy, TURF, SIM, SPIM, HILO, AV scan, fluorescence fluctuation super resolution microscopy, live cell imaging, single particle colocalization, and so on and so forth. Okay, so so this paper is you can download it from this QR code, and so the impact of this technology might be very high to the scientific community, because in principle, it allows to explore the nanoscales without the need of having access to expensive instruments. Then after, we have the possibility to support to advance the field of super resolution microscopy in Latin America. So here you might want to check this QR code to know what we are doing in terms of education. And we are also developing plugins for computational super resolution microscopy, we have released the Napari super resolution microscopy plugin that also have MESSER embedded. And here, for example, is the ImageJ plugin of MESSER. So you can download it and install it in MESSER. Also, we have here the Napari super resolution plugin. So the technology is already available to everybody. And so we are reading, so all of these are available in the original paper, but we are also writing a paper. We are about to put it in preprints about the tools. And in this case, it's more or less about a guide, what should do and what shouldn't do. For example, when we speak about parameters, so there is a single parameter which is very important, which is how big is the PSF in your data? So we need to compute more or less the spread of the PSF. And that is computed through the full width at half maximum of the PSF. And in this paper, we will discuss what happened if you put this value wrong. For example, if you put the value very, very short, you will underestimate the PSF, and then you will end up splitting information that shouldn't be splitted. So if you put this value bigger, then you will gain higher resolution, but you will not get the optimal. So so we give some recommendations to compute the full width half maximum of the PSF to get the maximal information out of your data. Okay, so with this, I'm about to finish. I will just tell you what is next. So, for example, this is some technology which is called image-based flow cytometry that combines flow cytometry with fluorescence microscopy. And this technology is limited by resolution because it uses air optics. So the resolution of the images is about 600 nanometers. And if you want to really use this technology to explore the subcellular scales, you won't get much. But what we have done with MESSER is that we have extended the resolution of this technology. And in this case, what I'm showing here is an sperm which was collected, an image of an sperm which was collected with this technology. And what we are seeing here is the flagellar width. And as you can see, we can really overcome the resolution limit in this technology and solve questions asking to hundreds of thousands of images simultaneously by combining imaging flow cytometry and super resolution microscopy in single images. So we can also download data from the repositories that are out there to go with this concept of data reuse. And here, for example, I'm just showing you some data from that was downloaded by one of the students of the lab. And in this case, this is the data, and this is how much we can increase the resolution of that data. So then, instead of asking questions about the presence or absence of markers, we can make questions about the subcellular localization. Okay, and more or less this is the kind of thing. So MESSER might open the possibility to do questions at the nanoscales with data that has been collected by other labs. So thank you. So this is my data, and if you end up doing something with MESSER, just use the hashtag MESSERit. Thank you so much.
That was a great talk, Ivan. Thank you so much. We have a question in the chat. Is the mean shift always perfectly symmetrical?
I mean, the operator is symmetrical, fully symmetrical. So the initial description, the initial implementation that we did, which is in the seminal paper, so the searching window is a square. So that makes it completely symmetrical. But what it is not symmetrical is the information. And that's why the mean shift works, because it computes the shift, it computes the deviation, it computes the local curvature of the information. So meaning that if you have a distribution which has aberrations, which is not symmetrical, in any case, we'll find the source of information. And that is what we are looking in fluorescence microscopy, where the information comes, which is actually from the fluorophores. Did I answer the question?
Yeah. And I think if anyone else has a question, you can go ahead and raise the hand or just unmute yourself and ask a question for Ivan. So I actually had a question about the IFC. Is that going to be available as like a plugin? Because you showed the idea software. Is it possible to do it like with an ImageJ, or are there plugins there? So what's the future of that?
Well, the plugin is already available. So in the NatComs paper, we publish the codes in Python, the codes in R, the codes in MATLAB, the plugin in Fiji, and now we have developed the plugin in Napari. So if you just ask for Napari SuperRes, you will have access to measure, but also to another super resolution methods. So in this project, you are trying to gather all the computational tools that do super resolution microscopy, just to have access to all of them to the people, and for the people to determine what would be the optimal for them. So it is available for everybody.
That's great. I like how you have it on like every platform and it's available. That's a real open access right there. We have another question. Aside from oversampling and using confocal microscopy, what else do you need using the MSSR application?
Actually, nothing else. That's it. So initially, so the most important parameter is knowing the size of the PSF, because that defines the searching window for the measure principle. After that, everything goes to, and let me give you an example. Let's imagine that you have collected an image which has perfect sampling in terms of Nyquist, that have, let's say, a pixel size of 100 nanometers. Then you might end up having like 4 to 5 pixels per point spread function. But after you do measure, after you measure it, you increase the resolution 2 times. Then you will not be in the perfect sampling. Measure have the possibility to increase the resolution much more. So then you might need to do interpolation. That is the other parameter that we have in the software. So what I recommend you, always use an interpolation of 3 or even more. Because then we split the pixels more and more times to compute and to oversample the space in order for you to get the maximum resolution provided by measure. So the other parameter is amplification, which is the degree of interpolation. Put a value more than 3, and then you will get the maximum resolution. There is another parameter, which is the order. Order 0 is using the first Mexican hat. Order 1 using the second Mexican hat, and so on and so forth. Depending on how much resolution you want to have, you use higher orders, but you will lose information. So I recommend to use a measure order of 0 or 1. So having these ideas in mind, now we are working in a new version of measure, which might be parameter-free. Just imagine to have parameter-free super resolution microscopy for everyone, for every technology, including other technologies like electron microscopy and so on and so forth. We are working on that.
Great, thank you. We have another question. We'll get this one. This will be your last one. Some of our confocal objective lenses that oddly shape PSF, sort of banana shape, does that affect method?
Yes, that question is really interesting. Not for the case of 2D imaging, but it is much more interesting for 3D imaging, in which you can you want to get in, like a 3D representation of your information, and in the information is banana shaped or deformed, then you might want to correct for that deformation or use that information to localize information in 3D. In any case, our idea is the following. What MESSER is trying to do is to identify the source of information. If it has the shape of banana, or if it has the shape of an egg, we don't care. We want to really go and identify the source of information. What we have in mind, but we haven't tested, we are working that. Indeed, I'm doing a small internship to do some of these experiments, is how much we can achieve using MESSER in 3D. Whether we can go for computational adaptive optics, we don't know. But if you want to share those banana-shaped PSF with me, it will be great, because then we can really test MESSER 3D on those.
Perfect. Well, thank you so much. I appreciate the talk today. It was fabulous. And I think if we have your email, if anyone wants to reach out for Adhan for more questions.
Yes, I'm going to put my data again. And I'm just starting this movement of MESSER it.
I love it.
Yeah, you know, because I've seen that there is now labs that are using it and making publication. I'm starting seeing the preprints just coming out there. So if you end up doing even some artistic information or representation with MESSER, just tag it as MESSER it. So this is my email, my Twitter. You can see a lot of things that we have done with MESSER there. And also look at the Mexican Bioimaging Workshops program if you want to have access to education in Mexico.
Awesome. Thank you. And I love the image.
Oh, yeah. I use DALI, artificial intelligence. To
Nice.
That's amazing.
That is perfect. All right. Well, I think this concludes the end of our today's speakers series. So let's all give Adhan a hand.